So, all right. So next up, we have Satpal Virdee from the University of Dundee in the UK. Received his master's in chemistry from the University of Surrey in the UK, and got his PhD in 2009 in Birkbeck College, London, under the mentoring of Gabriel Waxman. There, he studied SH2 domains using techniques such as X-ray crystallography and biophysical analysis. It's also the time in which he became interested in genetic code expansion through collaboration with Derek McMillan and developed expertise in the area of chemical protein synthesis. He then joined Jason Chin's lab at the MRC in Cambridge, where he pioneered the genetically directed synthesis of atypical ubiquitin chains and ubiquitin conjugates. Working closely with David Commander, he solved the crystal structure for List6 linked ubiquitin chain and identified a D-ubiquitinase, which is specific for atypical linkages. From there, he became the MRC program leader and personal chair in chemical biology, MRC protein phosphorylation and ubiquitination unit at the School of Life Sciences at the University of Dundee, where he is now. His research focuses on all things ubiquitin and has a keen interest in chemical biology of E3 ligases and their ability to be targeted with drugs and developing those drugs for novel therapeutics. Also interested in a subset of E3 ligases that perform non-lysine ubiquitination. With all of this research, there's been a major push in genetic code expansion-based tool development. And recently, Satpal has founded a company, Outrun Therapeutics, which is a fabulous name, and I'm curious as to how you were able to keep that one. It hasn't been taken. And it's focused on therapies that target the ubiquitin system. He's won several awards along the way, which I won't go into all of them, but because I think we'll be here for a bit. But with that, I'd like to welcome Satpal.
All right, thanks, John. So yeah, as John mentioned, we're interested in the ubiquitin system and E3 ligases, and this project is a little bit tangential to what we normally do. We're normally interested in the mechanism and of unusual E3 ligases, but we kind of got sucked into the targeted protein degradation sort of buzz, and we felt that using genetic code expansion and combining it with bioorthogonal chemistry, it could actually address an important unmet need in the field. And I had a really enthusiastic student who really wanted to work in this area. So this is what I'm going to talk about today. Uh, so in targeted protein degradation, it's just a small component of a much broader and really exciting therapeutic modality known as induced proximity. So this involves bringing together two proteins in a cell that don't normally talk to one another, and this is achieved with a small molecule. So you can rewire the cell to do things it doesn't normally do under native conditions, and this is of course exemplified by targeted protein degradation, where a small molecule can bind an E3 ligase and redirect it to a substrate, triggering its degradation by the proteasome. And this is, as I'm sure you're all aware, this is, generating a lot of traction as a way of getting rid of disease-driving proteins, which have been difficult to target using conventional small molecule inhibition strategies. So there's still a lot of potential to unlock just within the targeted protein degradation space. One area being the needs to target more E3 ligases. So the number that can be hijacked with a small molecule is increasing all the time. But I think it would be fair to say that the E3 ligases VHL and CRBN have been the most productive. And if they're working well, why do you want to recruit another E3 ligase? And I think the most important answer to that question is this, because the mechanism relies on a cellular ligase as part of the therapeutic modality. One can actually harness biological specificity in a completely new way, and by that I mean, can we get tissue specific or disease specific degradation if we developed a PROTAC molecule or something like that that only targeted a ligase that was active or had much higher activity in the diseased tissue. So this is a really important area where discovering tissue selective ligases is important. And induced proximity, other examples of induced proximity include things like dub-tax, where you recruit an enzyme that deubiquitinates, and this has the inverse effect. Rather than driving degradation, it's a stabilizing effect, and you can envisage applying this to things like tumor suppressors, where you want more of these things. It's essentially, this can be applied to any writer and eraser enzyme system to basically rewire biology. And I would say it's not even limited to enzymes with writer and eraser functionality. Cellular signaling is brought about by proximity. Signaling nodes are created in cells by proteins complexes coming together. So even using this induced proximity, it's conceivable to create new signaling nodes which aren't sort of directly harnessing uh, uh, writer and eraser activities. And also another area I'm really excited about with induced proximity is using it as a basic research tool. Where a pressing problem is understanding protein function and being able to, induce the proximity of proteins of interest to specific sites could actually be a very complementary way to understand the function of poorly understood proteins. So bringing proteins together has lots of utilities, but it doesn't always work just by simply bringing the two proteins together. If you've got an E3 ligase, the catalytic site needs to be in such an orientation where it can actually put the ubiquitin on the substrate, right? And this is one of the bottlenecks to really sort of unlocking new E3 ligases and I would say unlocking the exciting potential of induced proximity therapeutics more broadly. So it's a bit of a sort of chicken and the egg scenario, this problem, because to really assess induced proximity, whether that's actually generating a productive ternary complex, you need to develop a ligand in the first place, which is costly. It can take a long time, and then you elaborate it into a bifunctional proximity inducer, and then one might find it doesn't actually drive the therapeutic effect. You don't get ubiquitination and degradation of your target. There are tools out there that address this in part. These are genetic methods where you can tag proteins of interest with complementary binding proteins, for example, GFP and a GFP nanobody. This is the basis of an AdProm system developed by one of my colleagues here, Gopal Sapkota. And there are also other systems like DTAG, etc., where you can actually conditionally induce the proximity with a small molecule. But a caveat with all of these approaches is that they require a protein tag, which is placed at the N or the C terminus of the protein, and that drives ternary complex formation, which might not have any relevance to how a small molecule would induce the ternary complex, which of course would bind internally of an untagged protein in a cell or in a patient. So we thought, can we combine genetic code expansion with ultra-fast bioorthogonal chemistry to address this question? So this diagram here shows just a couple of examples of an unproductive, a hypothetical unproductive, and a couple of unproductive ternary complexes here, and this is a productive one. And you can see it really depends where you hook, where your molecule hooks onto the protein. That really dictates the ternary complex. So we thought, can we combine genetic code expansion with bioorthogonal chemistry to, it's not a drug, it's not a therapeutic method, but we were thinking, could this be a very effective way of assessing productive ternary complexes in mammalian cells? And we weren't the only ones who had this idea. We published this last year, and Ryan Mel's lab published something similar. Also, Ed Tate's lab and the Dieter's lab also published something like an inverse configuration before all of us. So we call our approach BPI technology, and it uses these what we call bioorthogonal proximity inducer probes, BPI probes. So to sensitize a specific site, so that's kind of modeling specific ligand binding, we genetically encode this bioorthogonally reactive amino acid carrying a dienophilic side chain, BCNK. And as a control, we also incorporate an unreactive Bok glycine. And BCNK undergoes ultra-fast Diels-Alder click type chemistry, similar to the click reactions that Toshi told us about, but this is much faster and doesn't require catalyst and only generates innocuous molecular oxygen as a byproduct. So as a proof of concept, we developed BPI probes containing a tetrazine group that would undergo rapid tetrazine ligation with the BCNK amino acid. And we just, in the first instance, we tethered these to this JQ1 ligand, which basically is a ligand developed by the Bradner lab, which targets BET family proteins, which are epigenetic readers and are important therapeutic targets. So this is how we synthesize the BPI probes. It's quite straightforward. We can buy this JQ1 molecule as an acid, and then we couple this this PEG species to it, and then activate this, and then react with an amino functionalized aryl tetrazine. So these types of structures can be, this synthesis can be applied to a whole range of established specific ligands for targets of interest, as long as there's a functionalizable site, which, and importantly, is that a permissive site? It's an area we can use as an exit vector without affecting activity. Then we can build BPI probes on these molecules, and that information is usually available because from the campaigns that developed the molecules in the first place. So we hadn't actually used mammalian genetic code expansion in the lab for quite a few years. So we sort of just redeveloped our own system, sort of taking bits and pieces of from the literature where people had shown sort of various things that can improve the efficiency for incorporating in mammalian cells. So we have 4 copies of the pyrolysyl amber suppressor tRNA. We introduced this U25C variant, which was identified by Abhishek Chatterjee, which corrects a mismatch, a base pairing mismatch in the pyrolysyl tRNA, presumably improves its stability. On the same plasmid, we have the pyrolysyl tRNA synthetase. For Bok incorporation, we use the wild type synthetase, and for BCNK, we use a double mutant that was originally reported by the Yokoyama lab. And then our target protein, the E3, the effector, is on a separate plasmid, and we use a piggyback plasmid, not necessarily for its ability to generate stable cell lines, more because of the fact that the target cassette is driven by a really strong EF1 alpha promoter. So we thought that would be important to get good expression levels to compensate for the perhaps modest amber suppression efficiency. And then we also introduced 2 fluorescent proteins to allow facile assessment of transfection efficiency, and also potentially to FACS sort, which is important sometimes, to select cell populations that are actually contain both plasmids, which are of course necessary for a functional GCE system. So as proof of concept, we just took the two known players, the E3 ligases. There are crystal structures available with their favored ligands bound, and we just chose this structures from the Chiuli lab, and we just selected amino acids that were solvent exposed in the periphery of the ligand binding site. And we did the same thing with cereblon E3 ligase and amino acids that were in proximity of lenalidomide, a common scaffold for cereblon-based degraders. So we, gratifyingly, using a fairly conventional dual transfection system, we could get good incorporation efficiency of the unnatural amino acids BCMK and Bok lysine at these multiple positions in VHL in mammalian cells. These are HEC-293 free cells. And also with cereblon at our selected sites, we could get satisfactory incorporation efficiency. So if we're going to use this system to sort of see what happens when we sensitize a specific effector protein of interest at a specific site, we need to be sure that we're actually measuring the recruitment resulting from clicking to that particular site. And amber suppression efficiency isn't exquisitely specific. There is read-through of native stop codons arising from that are present in the mRNA transcripts that are native to the cell. So we thought it would be important to gauge that and see what is the extent of read-through and what is the scope of this. And how much should we be worried about it? And as you can see here, we're just incorporating the clickable amino acid in mammalian cells, and then clicking on a fluorophore, and then resolving that lysate out on a gel and doing in-gel fluorescence. And you can see there is indeed read-through, as has been reported in the literature. But when we put in the VHL construct with the in-frame stop codon, in the back on top of that background, we get clear, robust labeling of the effector of interest containing the unnatural amino acid. So we had the system working. Rick in the lab streamlined the dual transfection procedure to get the times down, and nicely we could see very robust degradation of some of these BET proteins, in particular the short isoform of BRD4. And yeah, we saw degradation of the other BET family members as well, but to various extents. So the gel-based readouts or the blot-based readouts, they're far from ideal for assessing target protein degradation. So we wanted to move to a much more quantitative method. And so to do this, we used the high-bit system. So this basically involves taking a cell line, which we got from the Chiuli lab, a 293 cell line, where the BRD4 protein had a high-bit tag knocked in at its N-terminus at the endogenous loci. So what the high-bit system is, if you're not familiar with it, it's a small protein tag, and then you can lyse cells and add a kit, which basically adds a complementary part of the high-bit tag, and you basically complement a split luciferase, and then you can add as part of the kit and the mix. There's a luciferase substrate, so you can use luminescence as a way of quantitating the levels of your protein. Of course, using that system, you're not using the gel, the blots, to confirm that you've got your components present in the cell. So this is where the M-cherry and the EGFP and the MK2 proved to be very valuable, because we can quickly check that the cells have been, the transfection efficiency work was satisfactory before we move to the high-bit analysis, which is all done in a plate format using a plate reader. So there were some important controls to do. So I should mention that there's background incorporation. So we thought it's important to check, can any of those misincorporated proteins, they could be E3 ligases for all we know, can they actually drive degradation? And because we need to assess that. So I think it's important to do it this way. So we took cells containing all of the machinery, but the VHL didn't contain, the E3 didn't contain of interest, didn't contain a stop codon. And then we added, so we had that background incorporation of BCMK, and then we did a dose response of the BPI probe. It's important to test multiple concentrations, because with these bifunctional proximity systems, there's something known as the hook effect, whereas you might not actually see degradation or at a particular concentration, because each component is saturated, so you don't get the proximity induction. And the concentration that hooks, if you like, for a given protein can vary. So hence it's important to do this control, we feel, using a range of BPI probe concentrations. Also, when we incorporate bot glycine, we don't see any degradation of BRD4 either. So these signals, I should say, correspond to the BRD4 levels, endogenous BRD4 levels with the high bit tag. So bot glycine doesn't, when we have that there and add the probe, we don't get degradation. So it's not an artifact of any degradation won't be an artifact of the general incorporation of an unnatural amino acid. So nicely, when we actually express VHL with the BCMK at position 67, we get nice degradation. We thought maybe this is limited by genetic mosaicity because this is a transient transfection. We're degrading the endogenous protein, so the degradation efficiency would be limited by the percentage of cells that actually contain both plasmids. So we addressed that later, but this just shows that the degradation was dependent on the proteasome, and it was also dependent on cullen ligase activity, which is what VHL is. We inhibit a protein, the conjugation of NED8 to the cullens, which activates their ligase activity. So we can get degradation at other sites as well. And this is how we can test whether the genetic mosaicity is actually presenting a barrier to the degradation efficiency. Here we've done FACS sorting against the EGFP and the MK, so we can obtain a pool of cells that we are confident all express the genetic code expansion machinery. And in this instance, when we have those sorted cells, we can get a Dmax, the maximum level of degradation of in excess of 90%. Remarkably, the DC50s, we're getting the concentration where you get 50% degradation is in the sort of low nanomolar region, which is comparable to the actual, normal PROTACs, if you like, optimized normal PROTACs for this system. So we can test multiple sites in VHL and can find that some sites don't work, some sites do. It seems the data suggests that actually the exit vector on the known VHL ligand, which has been used for PROTACs, is probably optimal because the best site was positioned near to that exit vector position. But our data suggests that other sites or other exit vectors on that VHL ligand could be used to generate productive degraders. We can also do this with cereblon as well and get decent degradation. And this is dependent on the proteasome and also on this NED8 cullen ligase activation pathway, which is they're both important controls. So once we'd had the system established, we thought, can we use it to test a completely E3 independent effector, sort of moving towards the idea of using it to test induced proximity more broadly. So E2 conjugating enzymes, which operate upstream of the E3s, they can actually transfer directly to substrates, potentially in an E3 independent manner under certain conditions. So we tested this ability, and people have shown this. Dan and Mora has shown that you can recruit an E2 directly to induce target protein degradation. So what we found here is that we do get some degradation. The degradation efficiency wasn't great. What was really cool and interesting is actually we can actually trap the monoubiquitinated BRD4 protein here. You can see this upper band, but you can see it doesn't seem to serve as a very efficient degradation signal because we can actually catch it and see it. So yeah, maybe that the missing factors that are required to really extend the chain on that, which the E2 can't do, and that's necessary to drive degradation. We get really efficient click. I find this amazing. So you can see this gel shift here, right? This is the E2. This is after adding the 1 micromolar, or it might have been 2 micromolar. No, it's actually only 500 nanomolar in this experiment BPI probe. And you can see after 1 hour, we get complete upshift in the band corresponding to the E2, which corresponds to quantitative conjugation of of the BPI probe to the target. And we see this with all of the systems and sites we've looked at. We always see near quantitative conjugation at very low concentrations within an hour. And this is just quantitating that degradation for BRD3. That was the most degradation sensitive bet isoform. So with that, I'd just like to thank my lab, Ricardo, who some of you may have met. He came to the GCE workshop some time back. He really drove this project, and yeah, I'd just like to thank him. Thank you for your attention, and thank my funders. Don't know if we've got time. We've got time for some quick questions, hopefully.
Yeah, that was great. Thank you so much, Satpal. That was amazing. Um, as the host, I get to ask the first question. So you've had to work to get all this to work in mammalian cells. You could share some of
Yeah.
the hurdles that kind of, or how you've overcome some of the bottlenecks for efficiency in mammalian cells. I think maybe some of the listeners would like to hear
Yeah.
that. And you've stuck with transfection. Have you thought to move to a viral system to improve maybe not overall expression, but expression over the
Yeah.
majority of the cells?
Yeah. So yeah, I mean, we're very happy with the system. We haven't benchmarked it against other reported systems that are out there. So I couldn't really comment on how well it works, but you know, it certainly works a lot better than an early generation plasmid system we inherited shortly when I started my lab here. I think in particular, something I didn't, I think I forgot to mention that we used the nuclear export sequence that on the pyrrolysine synthetase, which Ed Lemke reported. I think that's really important for boosting efficiency in mammalian cells because he showed that without that, there's actually a cryptic nuclear localization sequence in the pyrrolysyl tRNA synthetase, and a massive portion of it gets sequestered in the nucleus. So having that export sequence, I think, makes a big difference. Um, yeah, Abhishek's tRNA variant. So we just kind of additively, picked improvements we came across in the literature, and we think we've got a pretty good system. But I would love to benchmark it, but I couldn't comment on how good ours is. And in terms of the stable cell lines, yeah, that's something we're working on. Efforts have been made in this area. I think there are some challenges though. When you integrate all of these components in a completely stable form, you get some unfortunate silencing of the
Yeah.
component mammalian cyst cells. So we're kind of trying to sort of hopefully not reinvent the wheel, but we are trying this ourselves. We thought, we're good to just apply our own strategy and ideas and see if we can get something that works. So we're not using viruses. They tend to have a limitation they can't package. Generally, the cassettes are too large. So we're not using viruses to do this. We're using like piggyback Sleeping Beauty system, and we're kind of switching to inducible um, uh, transcription of the synthetase and the tRNAs, and maybe that would be key to prevent this the silencing that occurs when they're being constitutively made.
I can add to that conversation a bit. So we just had
Yeah, yeah,
a
please.
study where if we put on the same plasmid a pull 2 and a pull 3 promoter, you actually get less expression of both because of the region in the nucleus in which those plasmids go to. And so if you think of pull 2 transcription and pull 3 transcription as buckets, if you don't have enough DNA that you put in, you only are going to fill each bucket up a little bit. And so splitting those, we like the tRNA from pull 2 any protein expression may help boost expression. Um, and so that's a system that we're also working on here in the center, and so we'll be
Right.
sharing when we get that going.
Right. Yeah, yeah. It would be good to compare notes.
Yeah, yeah. All of this stuff is hard,
Yeah. Yeah.
and sometimes good enough is good enough. Um, is there are there any other questions from the group? Oh, I actually do have one more question. So you've been focusing on hex cells and standard cultured cells. If you want to
Yeah.
move this into more specialized cells, or do you want to move your system into more specialized cells to look at ubiquitination, let's say in neurons or in cancer or where, the targets might be different for
Yeah.
therapeutics.
Yeah, I think it depends on the target you're looking at. Yeah, it's absolutely conceivable you're only going to get sort of a meaningful readout if you're doing the experiment in cells that recapitulate the character of the disease that you're interested in. So yeah, it's something we thought about, but we'll definitely try and explore once we've got a satisfactory system, just using standard 293 cells, a stable
Yep.
system. I think that's the major. I think that in itself would be a milestone, just to have a robust system that doesn't shut itself off.
Yeah, absolutely. Um, well, great. Thank you, Satpal. It was a great talk. Thank you both, Toshi and Satpal. And thank you all for joining us. And we look forward to seeing you in exactly a month from now.